Basically, the concept that we are going to explain or we are going to understand is that when we get the homologous chromosomes from the parents, homologous chromosomes, it basically means that we are getting two sets of 23, and each set has each chromosome exact copy of the other. These are called as homologous pairs or homologous chromosomes. So, in what sense they are they are homologous in size, shape, and genetic arrangement. It means that if okay, I would draw it again. It means that if I draw a chromosome, suppose that it is chromosome number one. This is chromosome number one that I'm drawing from any set. Let's say father set, and the other one is the chromosome number one from, let's say, mother set. Now, this particular chromosomes, uh, when we talk about the genes, basically, we say that genes are segments of DNA. And then we say that chromosomes uh, are actually the folded DNA. So what does that mean? That means that the genes are located on chromosomes. As they are located on DNA, they can also be found on chromosomes. Why am I saying that? See, when you fold the DNA, it would never change the fact that genes are still present. It would never change the fact that genes are still present, even if we fold the DNA into chromosomes. So the chromosomes are folded form of DNA. Chromosomes are folded form of DNA, and the genes are located on them. Every gene, now very important concepts are coming. Every gene has a dedicated position which no other gene can occupy. Position no other gene can occupy. It means that if it is the position number one, you can also say dedicated location as well, location or position. So if location number one is dedicated for chromosome for gene X, two is dedicated for Y, and three is dedicated for Z, and this is a chromosome number one from father's set, you will find the same arrangement in the chromosome number one on the mother set. We have established that yesterday because they are homologs. So you will find the same arrangement on the chromosome number one on the mother's and father's set. Now, next, remember that no other gene can occupy, meaning that the position number 1, 2, and 3 can never be occupied. The position number 1, 2, and 3 on chromosome number 1 are permanently occupied by these three genes. Permanently. You cannot change that fact. Okay. So that is why the location of a gene on a particular chromosome is called as locus. By from location, we say it locus. So chromosome number one, locus number one. It means we are talking about gene X. Chromosome number one, locus number two. It means we are talking about gene Y. Chromosome number one, locus number three, we are talking about gene Z. We are not talking about any other chromosome, any other gene, not possible, because these three are dedicated locus, or the plural is loci for these genes. So similarly, we'll be finding the same genes at same loci on the mother's chromosome. 1, 2, 3, X, Y, Z. Same genes on same loci on the mother's chromosome. So that is basically the idea of locus or loci. Now, A question can be asked that if both mothers, both mothers and fathers, mothers and fathers genetic makeup is so similar, is so similar, how the variety is possible in human beings. Like obviously, we are saying we are saying that the homologous chromosomes means the chromosomes are not changing. Am I right?
Yeah.
Second, we are saying that genetic arrangement is similar. It means there is no change in genetic arrangement. Second point. Third, we are saying that the the loci are dedicated. Means that the locus would never change. The locus would never change, and the gene would be at its permanent location or position. So, how is it possible that the variety would come? I told you three different sources of variety yesterday. But the thing is, if the genes are staying permanent, if the genes are staying permanent, how is it possible that the variety would appear? For that purpose, you need to understand something very clearly. I would just get away from the current topic very slightly and get back to it. See, when we talk about a gene, when we talk about a gene, when we talk about uh, a gene has a sequence of nucleotides, particular sequence of nucleotides. A gene has a particular sequence of nucleotides. Okay. So if this sequence changes, if this sequence changes, the resultant protein would also change. Would also change. That may or may not result. That may or may not result very you can say that may or may not come out in the favor of the organism, may not come out in the favor of the organism. This thing is called as, this particular change in the nucleotide sequence is called as mutation. Sometimes the DNA is like this: A, G, C, T, T, C, G, A. Now, if a change appears, A, G, C, T, C, G, a change appears, and instead of the original DNA, which is a normal one, it changes into AGCA instead of AGCT. Now, this would be considered as a mutant DNA. This would be considered as the mutant DNA. Remember that this mutant DNA, as I have said, it may or may not come out in favor. This mutation may be useful or may be dangerous. It could be dangerous. Then it means that the life of that organism is at stake. Is at stake. If it is useful, it will make the organism even fitter. Sometimes the change um, does not matter that much. Does not matter that much. Uh, Let me tell you. Suppose that there was a blood group A. Blood group A. A change came in the gene of blood group, and it suddenly became blood group O. Now this change is not uh, this change is not increasing the fitness. On the other hand, it is not putting the life of the organism at stake. You getting it? This kind of change only brings variety. It is not putting the organism's life at stake, or it would not make it even more fitter. It's just a change that appears. So the mutation can result in these kind of things. So it means now please focus here. It means that the normal gene can be there in the DNA. Can be there in the DNA. But the mutant gene or mutant DNA can also be a mutant DNA. Let's say this is the first category, this is the second category, and this is the third category. Mutant DNA or mutant gene of first or second category can also exist in the human being DNA, in human DNA. Like, no problem in that. If the mutation is useful, then obviously we would love to have that mutation in us. And if the mutation is not useful and as well as not harmful, there's no problem. We can keep it. Like my blood group is O positive. So it doesn't matter. It is a fact proven by the scientists that original blood group was A, but later it changed to O due to mutation. So I have that allele. It doesn't affect me. It doesn't affect me. So that means that is completely okay to have the alternate form. So it means that a gene can have, now let's come back to the, a gene can have multiple forms. A gene can have multiple forms, and these multiple forms of genes are called as alleles, alleles. How do we represent these forms? Suppose that the very early gene was represented as A. The mutation came and the other allele is represented as small a. The original gene was represented as B, but a mutation came. The M is representing the mutation. A mutation appeared, and the same B is now present in an alternate form B. So you would say that A is an alternate form of A. Or you can also write A is an allele of A. B is an alternate form of B, or small b is an allele of capital B. Now that is the main thing. That is how. Now let's get back to the concept. We are trying to answer, if both mothers and fathers' genetic makeup is so similar, how the variety is possible in human beings. See what happens, that when you get the chromosomes, let's suppose, again, let's draw the chromosome number 1 from father and chromosome number 1 from mother. This, suppose that this is the chromosome number 1 from father and this is from mother. This is the first locus, second locus, third locus. Same, first locus, second locus, third locus. These are chromosome number 1. Now, suppose you got this chromosome number 1 from your father and you have a gene A over here. This locus is dedicated for gene A. It means that when you would get it from your from the mother, obviously the locus 1 of chromosome number 1 is dedicated for gene A. So it would also have A. Suppose that the locus 2 of chromosome number 1 is dedicated for gene B. The locus 2 of the chromosome number 1 is dedicated for gene B, no doubt, but the alternate form, the alternate form or allele can occupy the locus. So it means that there could be a small b over here as an alternate form. You may have got a small b from the mother and a capital B from the father. I'm not right now telling that small b is more fitter and capital B is less fitter, or vice versa. But if if two forms of allele are existing, if two forms of alleles are existing, any one can appear at the locus. Can appear at the locus. So it means that now let's say the third locus is dedicated for gene D. You get a capital D from your mother, but you get a small d from your father. Let's create another situation. Locus number 4 of chromosome number 1. The 4 is dedicated for M, gene M, suppose. You get a small m from your father and a small m from your mother. Now, this is what brings the variety. This is actually exchanged during the meiosis, remember. This is actually exchanged. So that is why we say that no doubt it doesn't change the fact they are still homologs. They are still homologs. It is not changing the fact. But obviously, now we are saying that gene type is changing. Yes, it is a fact that in the homologs, the gene type can change. Or I would say gene form can change. I would not write gene type. Gene form can change. So in homologs, a gene form can change, but they are still homologs. Why? Because they are still same in size, shape, and genetic arrangement. Did you get all this?
Yeah, I got it.
Awesome. Okay. Uh, during all this information, we have one more idea. We have two genes. We have two genes for each character. For each character. One we get from father, and the other we get from mother. When we get from father and we get from mother, it means that suppose we have got capital A from mother and capital A from father. So we can write it AA. We can get capital B from one parent and small b from other parent. We do not have to draw chromosome every time. We can just write it BB. We are getting both recessive from our parents. We do not need to draw them every time on the chromosomes. We can write them MM. Okay, now, uh, all right. Another very important point that actually there is a relation between these genes. The chromosome number one, position one, the gene number one of chromosome number one from father's set is also called as allele. Allele, basically these A and A are alleles, B and B are alleles, D and D are alleles, M and M are alleles. What is the meaning of this? Basically, when we get one allele from our father and one allele from our mother, then both of those alleles, both of those genes are alleles to each other. What does it mean? It means if I have this chromosome, I have this like G over here and small g over here. I would say, let's suppose this is chromosome number 10, this is chromosome number 10. Then what makes them, that makes them homologs. These are homologs pairs because their number is similar, their number is same, they are homologs. So the definition of an allele can also change. As I said, the allele, the multiple forms of a gene are called as alleles. Or we can also write the allele is an alternate form of a gene. Form of a gene. Similarly, I can also write the definition of allele as a partner of a gene, partner of a gene present at the same locus, at the same locus of homologous chromosome. Just look at the G and G. The capital G is present at the same locus as small g on homologs. So there can be three definitions. One, multiple forms of a gene. Second, an alternate form of a gene. Third, a partner of a gene present at the same locus of homologous chromosome. Got it?
Yeah.
So it means that every gene, every gene we get from parents, we get from parents has an allele. But the reality is the allele can be same. A is an allele to A. The alleles can be same if we get both alleles, both same alleles from our parents. Our parents. Meaning that, meaning that uh, we can get A from father and we can get A from mother. Or we can get small a from father and we can get small a from mother. Given that, suppose A is the gene, so the allele can have this allele can be same. Alleles can be same. So, but at the same time, alleles can have a different combination. Can have a different combination. Alleles can have a different combination. Uh, we get small a from father or capital A from mother, or maybe other way around. So we use a terminology for this. We use technical terminologies for this. Combination of alleles. Combination of alleles is called as the genotype. In the language of genetics, the combination of alleles is called as the genotype. A genotype, in a genotype, both alleles can be same. If both alleles are same, we would give it a terminology of homozygous. Obviously, I'm saying genotype means combination of alleles we get from parents. If we are getting both same alleles from parents, then to represent them in the words, we would say they are homozygous. And if we are getting both different alleles from parents, we say that they are heterozygous. Like capital A, small a. This genotype is called as heterozygous. About the homozygous, we have an exception. We know that homozygous can be like this: capital A, or small a, small a. Are both alleles same?
No, they're different.
Like, no. Capital A, small a. Like they are, the capital A is an allele to capital A, and small a is an allele to small a. So, like, are they same or different? I'm not talking about these two. I'm talking about these
Oh,
two.
yeah, like uppercase A is the same and lowercase A is the same.
Obviously. So this is one genotype. You would call this one as homozygous, this one is homozygous, but the thing is that you have to differentiate them. So whenever you say that you have both uppercase A, you would call homozygous dominant. Homozygous mean both are same. In what form? In dominant form. And if they are lowercase, you would call it homozygous recessive. You would call it homozygous recessive. So basically, what this all means, it all means that if you get both capital alleles, this is a child is getting both capital alleles from the parent. That would be, now you would call the child's genotype for gene A. For gene A, the child's genotype is homozygous dominant for gene A. If the child gets one capital A and one small a, then for gene A, the capital A, small a, then the uh, genotype for gene A is heterozygous. And if the child gets both small alleles or both lowercase alleles from both parents, now no doubt they still are homozygous. They are again homozygous, but this time they are recessive. They are recessive. So, okay, is this clear?
Yes.
Now we have understood many terminologies here. We have got the idea about the homolog. We have got the idea about the alleles. We have got the idea about the genotype. Okay, these three terminologies are really important for the topic that is about to come, and that topic is, and yes, we have also got the idea about the mutation, but factors that cause the mutation, we haven't discussed that. We'll talk about that later because that is obviously this can be asked for 1 or 2 marks, but it would take us away from the topic flow. We are now going to enter into a very important topic that is called as inheritance. Inheritance basically represents the behavior of genes while getting inherited from one generation to the next. Okay, so now what is this? How are we going to understand this? If you remember the meiosis, I would like you to revise the thing in your mind. I said that meiosis separates the homologs. Ultimately, the meiosis separates the homologs. How? We have 46 in the beginning, then we have 23 after meiosis 1, but they are still in duplicated form. And then we do another meiosis 2, and in the end we have 23 chromosome. And 23 itself, let me write this information over here. A haploid cell always means that the homologs are no more together. No more together. The homologs have separated from each other. 23 have been separated from 23. Is that difficult to understand?
Oh, I understand.
Okay. The homologs have been separated, meaning that the gametes only have one part of homologs in each one of them. In each one of them. When the gametes fuse, when the gametes fuse, when the gametes fuse, by they I mean homologs, reunite. And what is the proof of that? The proof of that is if we have 23 here in a sperm and we have 23 here in the egg, they unite to form a zygote. It means 23 homologs are combining with the other 23 homologs, but now with different allelic combinations. With different allelic combinations than their mother and father. Different allelic combinations than their mother and father. So it means that if we want to suppose we have these two alleles. Okay, we have a gene A in a male. Obviously, this male would not have only one gene for that. The gene A would have its alternate form. Why are we so sure about it? Because it's very simple. We know that this male is a child of someone and he got one allele from his father and one allele from his mother. So obviously this male actually have capital A. Suppose it has both the dominant copies. It has both the dominant copies from his parents. First of all, name the genotype. How would you name the genotype? What is the name of this genotype?
Like of the capital A, what is the name of the genotype?
Yes, they both are capital. So how would you name the genotype?
So they're both dominant.
So how would
So
you name them?
homozygous dominant.
Well done. Well done. So this male has homozygous dominant. But having two alleles together means that the cell is diploid, because only in the diploid case you have homologs together. Now what would happen? You need to make the gametes of this male. Now what would this male do with these alleles? The AA would separate from each other into different sperms. Now this would be a different sperm, and this would go in the different sperm. This would go in the different sperm. This happens during meiosis. This happens during meiosis. So it means that um, tracking this behavior, tracking this behavior, we can we can draw the estimate, draw the estimate or probability. Oh, sorry. We can draw the estimate or probability of genotypes in the next child. In the next child. Okay, how? How? Please very closely see what I'm about to do. There is a male and female crossing, male and female crossing. The male has, suppose the male has a heterozygous genotype, capital A, small a, and a female has a, a homozygous recessive genotype. Now you know very well that in meiosis, the gametes are supposed to be separated. So these are, okay, these are the genotypes. Now the meiosis is going to occur, and when the meiosis occurs, both the alleles separate from each other. See, similarly in female, the alleles would separate from each other, and the circle, always remember this, these are gametes. The gametes are basically highlighted by drawing a circle around the alphabet. Drawing the circle around the alphabet. The alphabet. Now, now these are, after meiosis, these are sperms, and after meiosis, these are eggs. Obviously, if the male and female are crossing, then the sperm and egg are supposed to be fertilizing. They are supposed to be fusing. So we will draw an estimation square called as Punnett square. You can say that this is a probability square. The technical name is Punnett square.
Yeah.
The way of
It's the
using
one
it,
that you match the recessive and dominant, right? So recessive is on the
basically, yes, 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 exactly. You are exact at the bullseye right now. Basically, we are now trying to draw a probability that what is the probability of genotype in their next child. So let's say the sperms on one side. Remember, how would we use this square? We'll keep the sperms on one side, A, small a, and we'll keep the egg from the on the other side, small a, small a. Now, I'm constantly saying that we are drawing a probability. And if we are drawing a probability, then we need to know that that any sperm can fertilize any egg. Meaning, if this sperm fertilizes the lowercase one, the genotype of the next child would be heterozygous. If this sperm fertilizes this egg, the genotype of the next child would remain same. If this sperm fertilizes this egg, the genotype of the next child would be homozygous recessive. And if the same sperm fertilizes this egg, the result would be the same. So now what are we getting out of it? We are getting the idea that the genes, first thing we are saying that genes shuffle during meiosis. Genes shuffle and separate during meiosis. Separate into gametes, let's say, into gametes during meiosis. Second, they reunite in next generation, but that reunion is totally random. They may resemble, their genotype uh, may or may not resembling their parents. So there is what we call as inheritance patterns. The behavior or the patterns of transfer of alleles from one generation to the next. One more terminology I need to clear here, but first tell me, did you get this?
Yeah.
Next thing, why do we say some allele is dominant and the other allele as recessive? See, it is a fact that sometime the protein, the protein of one allele is more expressive or acceptable for the body as compared to the protein of the other allele. Or I can also define the dominant allele as um, an allele that produces such a protein that masks the effect of other protein, effect of other allele. Let me give you an idea about the recessive one. A recessive allele is the one who either does not produce a protein, does not produce a protein, or maybe produce an unacceptable or faulty protein or faulty protein. It may produces an unacceptable or faulty protein. So that is the meaning of a recessive allele. That is why we say some alleles are dominant and some alleles are recessive. Most of the time, we present the dominant ones with uppercase. But also remember that it is not necessary that dominant ones are normal. Maybe our syllabus would give us the idea that dominant ones are normal, but it is not true. So now the thing is that the alleles would define your appearance. Appearance is sometimes also called as the phenotype. Let's say we have there is a character. We say that hemoglobin protein. Hemoglobin protein. It has two forms. One form is normal, and the other form is sickled. Normal form and sickled form. If the hemoglobin is normal, it tells us that genotype is normal. And if the hemoglobin is not normal, it tells us that genotype is not in our favor or normal. So you can also say that actually now this normal, and what are these normals and sickle ones? These are phenotypes. These are phenotypes. The phenotype basically is a result of performance of genotype. How the genotype is performing would be expressed by the phenotype. Let's say if I say that a student is getting good results, what does it prove? What does it prove? That the student studied properly, maybe, right?
Yeah. Yeah.
The student studied properly. He got good practice material. He got good teachers. Anything. So if a person is getting good grades, that is a proof that the person may have got these factors at their best: the teachers, the notes, the test materials, the past paper questions. So that is obviously no one saw that when the student was preparing, while he was like locked in his room. No one saw that what that student is doing, how is that student preparing. But when the result came, everyone noticed that okay, he was such a good child. He used to prepare a lot. He used to lock in his study for 6 hours, for 8 hours, for 12 hours, etc. So people normally assume that that person must be studying very hard or good. So the phenotype is a performance proof of the genotype. That is basically the point here. Okay, so now let's suppose that mm, let's see different variable phenotypes. Let's talk about variable phenotypes. Um, let's say there is a phenotype of a character earlobe. Earlobe. Earlobe is a trait or a character. So um, earlobe has two variations, two phenotypes basically. Remember, earlobe is a trait. It has two phenotypes. One is attached, the other one is detached. You may have heard about these variations in the junior classes. So, attached or detached. Now, let's suppose that an allele a gene, suppose D, a gene D is the cause behind the attached earlobe, and its allele, its mutated form D, is responsible for detached earlobe. A gene D is responsible for attached earlobe, and its alternate mutated form small d is representing or responsible for the detached earlobe. Let's say there is parents, or we also call that p generation. Let's say the parents cross male and female, male and female, and the parents have a heterozygous genotypes, capital D small d cross capital D small d. Now, what do you think the phenotype of the parent would be?
Uh,
Given the fact that one dominant allele is enough in the genotype.
so it would be what, like capital P and lowercase p?
No, see, I'm saying that what would be the phenotype? I'm right now not talking about the genotype. The genotype is heterozygous. The
Yeah,
genotype is heterozygous.
yeah.
So, what would be the phenotype? In other sense, I would say these are the two possible phenotypes. These
So
are
is
the
the
two possible.
earlobe the phenotype?
Earlobe is the trait, is the character. Earlobe is the character. Its variable forms are phenotypes.
So attached and
So,
detached.
attached and detached are the phenotypes. So, my question is, what is the phenotype of the parent in case of earlobe? If I just ask you to look at the genotype very closely, what would be the phenotype of the parent in that case?
Attached.
Attached. Why? Because we are giving the fact that one dominant allele is enough. Both dominant allele, very good, but one dominant allele is enough. So now what should happen? You know very well. The meiosis should happen in both male and female. They are about to produce the next child, so they are going to produce the gametes. Now the meiosis would happen. They would be separating their alleles. Both male and female would be separating their alleles. These are now what? These are now gametes. These are now gametes. The very first generation that the parents produce is called as the filial or generation, or we also call it F1 generation. These are very important labels. Remember, the very first generation that is reproducing, that is crossing, is the P generation or parental generation. You have to represent the gametes as well. And filial generation is the one that is going to be produced. Now, what is your estimate? Let's say I ask you to estimate, to draw the probability. What is the probability of their next child being a next child having a detached earlobe? Detached earlobe. So let's say we draw the Punnett square. We put the sperm on the left vertically, capital D, small d. We put the eggs above horizontally, capital D, small d. If this sperm fertilizes this egg, then the child that is now you have to tell me what is the probability that the next child is going to have the attached or detached? Capital D, small d, capital D, small d, and small d, small d. Tell me the probability of the next child having an attached earlobe, and also tell me the probability for the detached earlobe.
So detached earlobe will be 25%
Well done. Well done. How can you say that?
because of the DD lowercase.
Yes, but try to name the genotype properly. It's homozygous recessive.
Mhm.
Yeah. So there is your answer is 100% right. How would you draw the probability for the attached earlobe? 75%?
Yeah, because what is it? Homozygous dominant.
This one is homozygous dominant. What about the rest of the two?
They're
Because
also
they are not same.
homozygous dominant.
No, they are not homozygous. They both are different.
So they're
So what?
heterozygous dominant.
Yes, 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 yes. No, no, no. Don't say dominant. There is nothing bad in saying that they are heterozygous dominant, but you can only say heterozygous. Just hold on a sec. Okay, so like this is one of the way to know the um, genotypes and phenotypes. Let's suppose if I say, okay, one more thing. Okay, um, we are basically supposed to be writing about the phenotypic ratio represented in Punnett square, represented in Punnett square. What are the phenotypes? Attached and ratio detached. So the total chances of the total number of chances are four. If you see, according to the Punnett square, the total number of chances are four. The 3 out of 4 is probability ratio for attached earlobe, and 1 out of 4 is the probability ratio for detached earlobe, or you can in other sense say 75% or 25%, but you must also know how to do this, because most of the time the CIs ask us the ratio, not the percentages. You got this? How I did this?
Yes.
Okay. Now they also can ask us the genotypic ratio. Genotypic ratio. What is the meaning of genotypic ratio? How many genotypes are shown in Punnett square, and how many times? How many times? So, how many genotypes are shown? Homozygous dominant, homozygous recessive, and heterozygous. So it means all the genotypes are shown here. So I would write homozygous dominant, ratio, heterozygous, ratio, homozygous, recessive. So, how many times homozygous dominant is shown in the Punnett square?
One time.
Once. So I would write 1. How many times
Yeah.
the homozygous recessive is shown?
One.
Only once? Yeah. So rest of the two are 2. So genotypic ratio for this cross, remember, it could change. Both parents are heterozygous. If both parents are heterozygous. Got it?
Yes.
Okay, when is your next class?
I don't have a class right now.
Sorry?
I don't have a class right now.
Okay, can I proceed the class till like for 13 more minutes?
Yeah.
Okay. So, so basically, the behavior of a gene, the behavior of a gene is basically found out in the inheritance topic. And we always do these kinds of crosses. Okay, now we have one more type of situation. That is very interesting that I'm about to say. Okay, let's say mm, a couple has four children, or let's say three children, two with attached earlobes, one with detached earlobes. Draw the genetic diagram to show the genotype of parents. Now this becomes interesting. Previously, we started from the parents and we moved forward. Now we know only the phenotypes of children and how two with attached earlobe, one with detached earlobe. Okay, so how can we estimate that? This is very interesting, yet a bit difficult. Okay, there are three children. Obviously, two children, they two have at least one dominant allele. Okay, can you guess how am I so sure about my this statement that two have at least one dominant allele?
Um, because it says two attached earlobe, right? So attached is dominant.
Well done. It means that two children have at least one dominant allele. But about the third, I am certain the third has the genotype of homozygous recessive. And how am I so sure about it? How am I so sure that the third one has the homozygous recessive genotype?
Third, third meaning like one third.
No, 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 not one third. Basically, the one with the detached earlobe, I'm talking about that one.
Oh, basically because detached is recessive.
Well done. The detached one is recessive, so it means it cannot have a dominant allele because he is already showing detached phenotype. So obviously DD. Okay. Now, all right. Be very careful what I'm trying to say. See, if this child is having two small d's, one small d this child got from one parent, isn't it?
Yes.
And one small allele, one lowercase allele, or one recessive allele, he got from another parent. So I can for sure say that their parents have at least one recessive. Why I can say that for sure? Because every child gets one allele from one parent and the other allele from other parent. So if this child is homozygous recessive, it means one he got from one parent and other he got from other parent. Okay, now you have got this point. Have you got this point?
Yeah.
Okay. So, but the fact is that their children are also showing dominant feature. And obviously, if children are showing dominant feature, where did they get that dominant feature? Obviously from parent. So at least one of their parent should have a dominant allele. Why? Only then there would be a chance that their children would be showing a dominant feature. If, like, a very obvious thing, let me give you a very obvious answer for this. See, if the parents don't have it, how the children can have it? It means that the both the parents have at least one recessive allele. We are 100% sure about this because this child is receiving one from one parent, other from the other parent, and two children are showing the dominant phenotype. It means either one or both of their parents have one dominant allele. Maybe one or both of their parents have one dominant allele, so their children genotypes can be justified. All right, now let's draw the cross. Let's separate the gametes. Now obviously we guessed that we were supposed to guess the genotype of the parent. This is our guess. Now we need to confirm that whether our guess is right or wrong. So let's put the sperm on one side and eggs on the upper side. D D D small d D small d small d small d. All right, now tell me, do does this Punnett square contain the probability for a detached earlobe?
Yeah.
Yes. But does this contain the probability for attached?
Yes.
Yes. So it means that it is a high chance that the parent genotype would be like this. There is a high chance. Always remember there are two ways of drawing a cross. One is from parents to offsprings, but the other way is to draw the cross in reverse. And we did the same here. We estimated that what would be the genotypes of the children, and then we drew a probability that only this could be the genotype of the parents. Is it understood?
Yes.
Okay. Now, basically, over here we are discussing what information. Actually, these informations are discussed here. Homozygous, as two identical homozygous breed together will be pure breeding. That I would explain later. These are secondary terminologies basically. What is a heterozygous? Dominant allele. What is the dominant one? Recessive allele. Okay, the pedigree ones are really important, and we'll be discussing that in one lesson, next lesson. Now look at this: 1-to-1 ratio and 3:1 ratio. This is something really interesting. Have we shown the 3:1 ratio?
Yes, we did. We did the
Yes, we
one
have
to
done.
two to one. Yeah.
Yeah, 1-to-1 is the genotypic. I'm talking about phenotypic ratio. We have shown 3:1. Basically, 3/4, 1/4 also is written as 3:1. Okay, so now, hmm, all right. So basically, next is I was about to tell you something. It just flashed off my mind. Yeah, basically, those terminologies. Okay, we'll discuss those terminologies later. I'm about to show you something, and I'll send that to you as well. Hold on. Worksheets, inheritance. Okay. Monohybrid practice. Yes. Look at this. This is a worksheet that I made, and most of the questions are these are mixed questions, O levels as well as IGCSE. So it would help you a lot. Now, the question in the question, there are three basic questions in this particular worksheet. You are supposed to be doing these